So, hi everybody. Today we have Mathieu Chevalley from ETH Zurich and GSK AI, who is presenting causal bench presentation, temporal graph learning reading group, obviously. And he's a PhD candidate at ETH Zurich and GSK AI, supervised by Professor Nikolai Meinshausen and Dr. Patrick Schwab, developing scalable machine learning methods for causal discovery from large-scale biological data. And yeah, with that, I hand over to you. With maybe one more question, do you prefer us to ask questions in the end or during your talk?
You can, yeah, you can interrupt me. That's fine.
Okay, cool. Thank you.
Great. Thank you for the introduction. Today I'm gonna talk about causal bench, which is the benchmark uh, for network inference from biological data. Uh, but maybe before diving into the benchmark itself, I wanted to give a bit of an introduction about causal inference, because in case not everyone is familiar with it. So the main goal of causal inference is to identify causes of some phenomenon. So, for example, in weather or climate, you can try to understand which variables affect the weather, the climate, what are the causal factors behind those weather phenomena. Same thing in biology, especially what is driving a disease, what are the cause, and then if you understand the cause, you can then target them. So maybe the main difference is that we're not only looking at prediction or association, but we want to understand what happens when then we affect and intervene on survival. So, for example, when we give a drug to someone. So, for example, like you have some gene A and some phenotype, and you observe this association. Here, maybe you know that gene A affect the phenotype, and you see it in the data. There seems to be some association between the two variables. But you may have another gene, B, that also looks like it's uh, uh, correlated with the phenotype, but actually it's driven by some confounders. And that's so for prediction, maybe that's not really important, the distinction between the two, but if you then want to intervene on a gene to change the phenotype, you will only observe like an effect on the intervention in the first case and not in the second case. So an overview, what's a bit maybe the difference with statistical modeling and probabilities. Uh, so yeah, in probabilistic modeling, you have a model, and from the model you can uh, do some prediction and predict outcomes, or you can go the other opposite way. You have some sampled observation, and you can feed to learn the parameters of your probabilistic model. So it's a bit similar in causal modeling. You may have a causal model. From the causal model, you can do causal reasoning, which goes further than predicting what you can observe, but can also predict the outcomes when you do interventions. Or in some causal model, you can also predict counterfactuals. So I observed an event, but what would have happened if, for example, I took another drug or didn't take a drug? But also the opposite way, from data, you may want to try to learn the causal model if it's not known a priori. So usually for causal modeling, we use also like graphs and graphical model, which are always directed graphs. So the directed edges uh, denotes the causal inference from one variable to the other. And then like usually also the graph is acyclic. So for example, you may have here, the season affects flu and hay fever. Flu affect muscle pain and congestion, but hay fever only affect congestion. And that's a way to represent this knowledge. What goes further with the causal model is that it's also like a generative model of your data. So what you can read from a causal graph is some independencies, a conditional independency you will see in the data, and also a factorization of your joint distribution. A bit more formally, so you assume you have some set of random variables that have a joint probability distribution, and that there is an underlying directed acyclic graph where the vertices are the random variables and the edges represent the causal relationships between variables. From the graph, then you can factorize the joint distribution based on the parent. So then really on the factorization, you see that a variable depends conditionally only on the parent in the graph. And also kind of what goes further that all the elements in the factorization are independent. So if you act on one mechanism, it doesn't affect the other one. So that's why it's not just any factorization of the joint distribution. Now looking a bit at like in general, let's say you assume there is an existing underlying graph that generates the samples you observe. Now, what you often want to do is that you don't know what's the true graph and you want to learn it from data. So learn the structure just from samples, which then allows you to have some understanding of phenomenon in your environment. But this is not such an easy task. For example, when you have what we call observational data, which is just you collected some data and didn't do anything else, you may not be able to find the true graph. So typically, if you just have two variables, even if you see an association, like here, you cannot differentiate whether there is a confounder, whether Y is the cause of X or X is the cause of Y. And in that case, we say that it forms an equivalence class where we cannot distinguish some graphs. But fortunately, if you have more than observational data and you have interventional data, so it's the same system, but you did an intervention in the system. So you set one of the variables to some value. For example, you give everyone the same drug, like in an RCT. Then you have more information because the new intervene data set is generated by a graph which is very close to the observational graph. Basically, when you intervene here on variable 2, it just removes the edges that goes to 2, but the rest of the graph is the same. And you can use this assumption that the graphs are very close between observational and interventional data sets to better learn the graph. And there are some results that the intervention can only improve your recovery of the true graph. Okay, so that was quick introduction about causality. So now I'm going to talk about the main topic, which is causal bench. So it's a large-scale benchmark for network inference from single-cell perturbation data. So what's the kind of the task in general? So you have some biological entities. Here it's mainly genes, and you assume that there is probably like what is called a gene regulatory network, which model what gene affect other genes or how they are causally related. And this can be really useful to understand what's the biological mechanisms that are in play. And if you understand the mechanism, you can better choose, for example, molecular or like a gene on which you want to intervene with a drug. But also nowadays, we have in biology, there are new measurement technologies and experimental technologies such that you can do intervention and more specifically, you can intervene on some gene and silence them or augment their activation. And you can observe then what's the effect of that on the other genes. And as I said before, theoretically, it should help learning a better gene network. And that's what we wanted to test. So the goal, before that, there was no real benchmark for this tag, especially given this interventional data and no way to compare different causal methods. So we wanted to create this benchmark using this quite large-scale single cell data and see, okay, does first existing method can already solve the task, and if not, what are ways to improve the result? And it also kind of test for causality because in theory, that's the perfect data and the perfect task for this type of models. So the contribution is obviously designing the benchmark, preprocessing, and having the code for this data, which is large scale. I mean, especially for causal task, it's pretty hard to have that many samples and interventional data also. We fixed all the evaluation procedure and data splits. Of course, we designed the quantitative evaluation, which is I'm going to explain, but it's not that straightforward in that case. And also we look at scaling characteristics in terms of number of samples, but also number of intervention. As I said before, more intervention should help learn a better graph, and we also wanted to test this characteristic. So to recap a bit, the problem here more targeted toward this gene domain. So we assume that there is some graph that would represent how genes are related to each other. We have observational data points where there was no intervention. We just observe how the genes are expressed in some type of cell. But then we also have some samples where one gene at a time was intervened. So here you intervene on gene B, and then you see the effect or which gene are affected. So if the graph is true, you would see that gene D changes in distribution, same with, for example, gene C, etc. And on the other hand, if you intervene on gene D, you would not observe an effect on the other genes. So now the goal is, given just the sample data, try to recover this underlying causal graph. So the data, as I said before, is quite high scale for causality. Usually people look at very small settings where you have maybe 20 fibers, but here you have genes up to 1,000 genes and a lot of samples, both on the observational but also in interventional setting. And we also have two cell types, so you can also see in different contexts how it behaves. Now the question is how to evaluate the output gene network of some method. It's not that straightforward because we don't exactly know the true gene network for those cell types. There is some prior knowledge. So one type of evaluation is this biological evaluation, where we pulled different sources of prior knowledge of gene interactions. So yeah, those are specialized biological databases. And then the second type of evaluation is a statistical evaluation, based only on the data that we have. So let's say there is a prediction of an edge between A and B. If it's actually a true edge in the graph, then we would expect that when we intervene on A, B should change in its distribution. And to measure that, we use a distributional distance. So in this case, the Wasserstein. And a higher distance means that there's a stronger effect. And yeah. Now, so it's kind of also like a measure of precision in the prediction. So if you take the average over all predicted edges, it should be high if you are close to the true graph, or you have high precision. Now there is also this kind of the opposite. So now, if you don't predict an interaction, so if there is no directed path from A to B, then you should not observe a change in distribution. And for all those like negative predictions, we can also test that this statistical test is not rejected, and then we calculate the false emission rate. And that's kind of the recall in that case. So there is, I'm going to show later, but there is like a trade-off between the two. So to show again, given the picture, so if we intervene on gene B or C because they are parent of D, then we observe changes in distribution that we can measure statistically. On the other hand, if there is an intervention on D, we should not expect a change in distribution for A, B, and C. So this is on simulated data to show this trade-off between the two metrics. So it's kind of like a precision recall trade-off. So of course, if you predict very few interactions you are very sure about, you may miss a lot of existing other interactions. But if you predict too much, probably including non-existing interactions. And those are the results, actually the first results we got when we did the first study of existing baselines. What you have to observe is that, basically none of existing methods work at all. So they're basically very noisy, and there is no differentiation between them. Maybe also what's more important is the one in pink are interventional method. So they are methods that use the interventional data, and in theory they should perform better. But as you can see, they basically perform the same as observational models. And if we look a bit more into this, so here we evaluate the interventional methods, and we vary the fraction of variables that are intervened. So, for example, if you have 20%, it means only 20% of the genes have an intervention. And we increase until 100%, which means all genes have an observed intervention. And what we would expect is that really this the performance or the metric goes up as you get more intervention. But as you can see, it's basically flat, which means, something is not right. So then I'm going to talk about this other related project which was inspired from the first results we got from the benchmark, where we organized a machine learning challenge to try to improve what was like the state of the art. So we organized this challenge within a workshop on machine learning for drug discovery at iClear. And yeah, we used the workshop. It was part of the workshop to have this challenge, and was a way to incentivize the community to focus on this important task and see if we can get improvements. So yeah, of course, we give them easy way to get into the problem, given the existing benchmark, the existing evaluation, also some starter baselines that they could use, and then the goal was for them to either tune existing method or propose completely new ones. And the task, as I said, so it's the same as the original one, is to predict a gene network given some interventional data. And as I said before, the main thing we observed in the first study was that, strangely, the interventional method did not perform better with more interventional data. So we really wanted to focus on that and incentivize new methods that really show an improvement with more interventions. So that's why we designed the evaluation metric around this. So we run the submitted models at different percentage of intervention, going from 25% to 100%, and then we compute the mean Wasserstein for each setting. And then we measure, so then it creates a curve for each models, and then we compute the AUC of this curve. And of course, higher AUC means it's really improving given more data, and yeah, that's what we wanted to see. And we tested on both cell lines. So now going straight to the result, as we can see, so all the methods at the top are methods that were proposed during the challenge. And when you see at the bottom what were the existing baselines, which were kind of flat lines in terms of improvement, here we really see what we wanted to see, which is more intervention gives better a better metric. But what's interesting is that it also translates not just to this evaluation, but also to the other evaluation that we had before. So this precision recall kind of trade-off on both biological and statistical evaluation. We see also a big improvement compared to what we had before. And yeah, this time we really see that those interventional methods work better than observational. Yeah, so quickly, the main also ideas behind those kind of winning methods. The idea is that they directly compare the distribution between observational and interventional to have like a set of potential candidates, and they combine that either with other models, or they do some also then filtering. But yeah, the main idea was that you should really compare this gene change in distribution when there is an intervention, and then create like a candidate set of interaction from that. Now, some limitation. Most of those methods only consider the interaction independently, even though you could have like number your whole interaction to maybe regularize your potential solution. And also they didn't really look at they don't differentiate between the direct and the mediated effect. Direct effect means there is really a direct edge between the two genes or variables. Mediated means that it goes through a path and that it's mediated by other variables. So yeah, in conclusion, I think benchmarking is definitely important, especially for maybe that case for biology, which is hard to get into if you just work on machine learning, just understanding the data, and yeah, and getting started. So having this benchmark really facilitate people getting into this and working on this problem. And also then it facilitates advancements with also the challenge really helped getting people interested in the problem and making rapid new new advancements. So yeah, if you're interested to know more, of course there is the paper about the benchmark, which is here the first one, where we describe the benchmark also to a more extensive study of existing method and look at more characteristics than I presented here. There's also a separate paper about the challenge where we describe the challenge, but also present the method that were proposed within the challenge. There's a GitHub repository, so of course the benchmark is public and free to use, should be easy to install and then try your method and compare to existing baselines. And also, if you're interested in this work, we did some follow-up to that. So based on the result and yeah, the new challenge method, we derive also new models for causality based on those ideas and formalized why they work and also did some theory around it. Yeah, that's it. From my side, happy to take questions.
Thank you very much. So if anybody has some questions, feel free to just ask them or type them in the chat, whatever you prefer. Maybe I have a question to start with. So you showed that in your competition, some of the methods submitted already quite improved over the baselines. What would you say is the way forward? Is it even better methods to like work on the current limitations, or is it more like even trickier tasks, even more benchmark tasks, or a combination?
I think they have improved already quite a lot. So yeah, from what I've been working on since then, they are hard to do better. But I think it's still possible to do better, especially let's say in the regime where you have some interventional data but very little that you maybe need to get more like modeling innovations. But then yeah, forward it's also looking at integrating other data set to do transfer learning, maybe also looking at time dependent data. Yeah.
And when you say time dependent data, is there already something like concrete that you have in mind or planned? Because that's of course interesting for us.
Yeah, I know. I mean, we are also thinking about it. The main issue is that yeah, there's not a lot of existing data set for gene expression at different time points. So it's more a question of the data set are not really there yet, especially for interventional. But I think it's already the technology is more or less here for that. So
Okay, cool. Thank you.
I,
Um,
I I was wondering a bit like are there other domains than like gene and biology where this could be applicable, or like what other kind of domains are working with this type of problems?
Yeah, that's a good question because yeah, indeed. So I would say that's why maybe causality method didn't look at this scale and also this interventional data because indeed there are not many domains where you have interventions, where you can do intervention, which is yeah, the main principle of causality is that it's hard to do the intervention and you're trying to predict in advance which one you should do. So for now, I would say the main application domains are either close to the healthcare and biology, where either you can do some experiments or you can have kind of synthetic experiments, or maybe some more closed models, or like a computer system where you can also intervene in the system easily, and then uh, that's also
I'm, I
possible.
was just
Yeah.
one. I was just wondering, right? Because you can think of interventions like in social experiments as well, right? Okay, how is COVID doing? Now we shut down a country. What happens to people's behaviors? Like, so, but maybe it's like what type of interventions are. It's also because I have no idea about this domain, right? So like what are some type of interventions that you do in this space?
Yeah, I mean, uh, those examples you gave are types of interventions, definitely. Uh, it's more, yeah, you could compare if you have two, I don't know, similar cities in one city, you could uh, enforce a mass mandate and the other one not, and that's an intervention. Uh,
So, so in the genomic space that you're looking at with GSK, it is kind of like different experiments with the genomes, or like what are the type of interventions?
so here in that case, it's so you observe gene expression, which is the abundance of each genes in a cell. And then what you can do is go into the cell and pick one gene and just suppress it such that it's not
Right.
activated in the cell anymore. So that's the intervention. And then you can observe how the cell is going to change. Uh,
All right.
but other type of intervention can be a chemical, a drug. Yeah.
Thank you.
Hey, thank you for the presentation. I just had like a very basic question. In your when you said you do interventions, do you do it on your observational data, or is it like a synthesized data set? I think it was on slide 6, I guess.
Okay. So, I mean, in general, you would assume that you mean here.
No, not here. I guess I think it's where you showed that where you like
This one.
compared the no, I think a few slides forward. Not this
Um,
one. I think you can go forward.
yeah, this
Yeah, I
one.
think this. Yeah, this one. Yeah,
Yeah.
19.
So, yeah, in biology, so it's the same population of cell, but it's not the same cells because uh, in the experimental uh, setting, once you observe the expression of the genes in one cell, it destroys the cell. But it's still comparable because those cells are like clones and their culture to be more or less similar, so they should have a similar distribution. Does that answer the question?
Okay. Yeah, thank you. So maybe I had like a follow-up question. So when you do interventions and observe that, let's say like G&T, it shows like a different behavior or something. Maybe I'm just expressing this in like very high level terms. How can you how can we ensure that the interventions that you did are like purely causal and not associational? Like, how can you ensure that? Like, yeah.
Yeah. So, I mean, that's the main idea is here. Here, given you do an intervention on B, you can't consider that it's not a random association, but if the only change between observational and interventional setting is the intervention on B, then you are sure that it's a causal relationship. But there can still be other confounders that create some association, like batch effects, co-culture in the same close, same tubes and stuff like this. So they can be confounders, but if you can normalize this away, then the only changes you see should be because of the intervention, which means it's like a causal relationship.
Okay, got it. Thank you.
Sure.
Okay, so if there are no more questions, I would say thank you very much for your presentation.
Sure,
And
thank you.
have a nice rest of the day.
Thanks. Bye, everyone.
Thank you.
Thank you.
Bye.
Bye.